So, welcome back. So, we have been discussing on the criteria upon which the classification of animals is made, isn't it? And we saw that there are different criteria starting from levels of or grades of organization or levels of organization. Then germ layers based upon which the eumetazoans were classified into diploblastica and triploblastica. We also saw symmetry, right? Radiata and bilateria. Again, eumetazoans were grouped into the radiata and bilateria based upon the symmetry, right? Now, coming to the coelom, we already discussed what is a coelom. Eucoelomates, we can see that based upon the presence and absence of coelom, we can have three kinds, right? Acoelomate, pseudocoelomate, and eucoelomate. In eucoelomate, annelids and arthropods had a hemocoel, isn't it? The coelom which is filled with blood-like fluid, right? Coming to the different kinds of eucoelom, it could be either schizocoelom or enterocoelom. Okay, in schizocoelom, what happens? We have already mentioned that in the eucoelomate groups, they have three separate cellular layers, and from the mesoderm arises the coelom, isn't it? And the coelom do have mesodermal lining. That is so specific about the eucoelomate groups. Okay, so in the case of schizocoelom, what happens is you have the ectoderm, you have this endoderm, right, and the mesodermal cells in between. A split appears in the mesoderm. Okay, that is what happens. A split or a band develops in the mesoderm, which enlarges to develop into the coelom. Okay, schizo means splitting. Okay, so this is how it happens. So a band or a split appears, it widens out, and it forms later the what you call coelom. Okay, and all around the coelom, it is lined with mesodermal cells, right? So that is what schizocoelom is all about. Now, what about enterocoelom? This is the you can see here, right? uh, the a pouch is developed. Okay, it's not pouch actually, it is what we call the outpushing is developed into the archentron. Okay, from uh, the in the blastula stage, outpushing is developed into the archentron, right? And then what happens, you can see here these outpushings. Okay, this is just an outpushing, okay, which is lined with mesodermal cells, right? You can see it is just an outpushing, it is not a cavity. Okay, so you can see here this is how it is. It continues it is, but it is pushed into the archentron. Okay, but later in the development, what happens? This outpushing, it gets separated from the lining. Okay, and it develops into a pouch-like structure, and this widens out and made together. You can see this has widened out. Okay, and this developed into the coelom. Fine, and this is known as enterocoelom, because what is enteron? Enteron is refers to the gut, isn't it? And here the gut is developed from the archentron, isn't it? Right? Okay, so this is what happens. Okay, so this is the enterocoelom. Now the next one is metamerism. Okay, so we can see all the organisms doesn't show the segmentation of the body. Okay, lower groups of organisms as well as mollusca and echinodermata, they have no metamerism. The body is not divided into segments. Okay, body is not divided into series of compartments along the anterior posterior axis. Okay, but metamerism can be found in higher groups. Okay, tapeworms show false metamerism. That is, they are pseudo metameric. This exhibits pseudo metamerism. True metamerism is found in uh, annelids, arthropods, and all the chordates. Okay, in annelids, you can see that externally and internally they are metamerically segmented annelids. Okay, what do you call it? Is a true metamerism. Okay, uh, the true metamerism, you can see it is exhibited by paired mesodermal somites in the embryos. Okay, and here you can see. Okay, coming back here, annelids, they are externally and internally segmented. Okay, outside and inside, you can see they are the whole body is divided into smaller segments. Okay, compartments, right? Each compartment being almost repeated. Okay, arthropoda, internally it is not segmented, but externally you can see, you may have seen the structure of arthropod, isn't it? If we take arthropod, it is like the head is there, okay, the thorax and the segmented what do you call abdomen, right? And if we see closely, the thorax is further divided into three. Okay, even though it is fused, we can see it is three segments fused together. It is in the case of insect I am talking about. Okay, similarly in other arthropods also, external segmentation is visible, but internally it needn't be segmented. Okay, it needn't be segmented always. Okay, now in the case of chordata, right, the higher groups. Okay, so there we can find it is the segmentation is purely internal, and that too during the embryonic stages. Okay, there is no external segmentation. Take for example human beings. Okay, there is no what you call segmentation externally. Now, internal segmentation, which was present in the early embryonic stages or embryonic stages, you can see that is still what you call reminiscence of that is still remaining in the adults, okay, or in the full-grown individuals, where you can see such kind of segmentation being present in repetitions of these being present in what you call rib arrangement or what muscles arrangement, etc. Okay, there is a slight kind of a metameric nature. Okay, but it is not pure, true. So it is only the internal at the embryonic stage. Fine. So they are all metameric segmented. I hope it is clear. Okay, the last part is based upon the how the mouth and the digestive tract is developed during the embryonic development. Okay, so based upon the process, the method by which the mouth is developed or digestive tract is developed, two kinds of like groups can be found. Okay, two groups can be found of bilateria, group bilateria. Okay, so based on the embryonic formation of mouth and digestive tract or anus, we can find two groups of bilateria. One is the protostome and the other is the deuterostome. Okay, or we can call the group as the protostomia and deuterostomia. Okay, in protostomes, what happens is the what you call the mouth is gastrular and it develops from the embryonic blastopore. Okay, so this is the blastopore. Okay, so this is embryonic. This is embryonic development. Okay, and this is the what you call cavity which is supposed to develop into the alimentary canal, the gut. Okay, and this is the blastopore. Okay, and in the later stages, what happens is it is this blastopore which develops into mouth. Okay, and so the first developed opening of the digestive tract in the case of protostome is the blastopore. Sorry, what you call mouth. Okay, and anus is developed from a pore which is developed later. Right, so mouth is developed earlier to the anus in the case of protostomes. Fine. Okay, thus protostomes are the animals in which mouth is formed first from the blastopore and anus is formed later from a second opening uh, away from the mouth. Clear? Okay. Now, how the name has come? Proto means first formed. Okay, or first protozoa, isn't it? So it is first. Stoma means mouth. Okay. So what is here actually? It is the first mouth we can say. Okay, mouth is formed first. Protostome. Okay, and this includes all the acoelomates, pseudocoelomates. We already mentioned that it is the bilateria which is divided into these two groups, isn't it? So in bilateria, you can see, right? A great bilateria, it is divided into three series, isn't it? Acoelomate, pseudocoelomate, and eucoelomate, right? And all the what you call acoelomates and pseudocoelomates, they have protostome kind of a development. Okay, and even some of the what you call eucoelomates also do have, for example, annelids, arthropods, and some minor phyla like Onychophora, etc. The Onychophora, okay, they do have what kind of a development? Protostome kind of a development of the digestive tract, okay, or mouth. Okay, now what happens in deuterostome? Okay, deuterostome, here we can see that the blastopore develops into the mouth. Sorry, what you call anus. Okay, blastopore develops into the anus and mouth is formed from another opening which appears later during the embryonic development. Okay, so deuterostome, deuteros means second. Okay, so deuterostome, here you can see that the anus is formed first from the blastopore. Okay, and mouth appears later from a second opening away from the anus. It is just the opposite of the deuterostome. Okay, so in protostome, blastopore becomes the mouth, and in deuterostome, mouth becomes the I mean, anus is formed from the blastopore. Okay, blastopore becomes a mouth here, and blastopore becomes the anus in deuterostomes. Clear? Okay. Now, what are the other differences between protostome and deuterostome? Here you can see, here a pouch is developed and it extends, isn't it? So in protostome, what kind of condition we can see? Okay, how it is actually. If we compare these two, we can see that there are yet another, yet like a very specific differentiation can be made between the protostome and deuterostome. Okay, one is protostomes, they have a spiral cleavage and determinate kind of development. Determinate kind of development in the sense each cell in the embryonic stage is already destined to become a specific part in the full grown individual. Okay, so if we remove a particular cell during the embryonic development, what will happen? That particular organ will be missing in the complete grown individual. Okay, on the other hand, this deuterostome, they have an indeterminate kind of a development, wherein the cells are not predetermined to become or predestined to become a particular part of the body. Okay, so even if we remove a cell, it is not going to affect the development of a fully grown individual. But if you remove a cell during the embryonic development of a protostome embryo, what will happen? It is going to cause a malformation. Okay, a part of the body, a structure, or some there will be some kind of a malformation. Okay. So this is regarding the protostome and deuterostome. I hope it is clear. Okay, fine, right. Thank you.